Another approach to anti-cytokines therapy is the development of hybrid protein containing cytokines receptor binding sites attached to immunoglobulin constant region. The only member of this class that has been approved for human use is etanercept or Enbrel. Enbrel consists of the intracellular domain of the type 2 tumor-necrosis factor receptor fused to the heavy chains constant region of IgG1. This fusion production protein can bind tumor-necrosis factor alpha, block its activity, and has a 4.8 days half-life in serum. However, about 16% of treated individuals develop antibodies to the tumor necrosis factor receptor 2 or moiety. Some of these antibodies may be able to bind to native receptor and activate signaling pathway, thus mimicking tumor necrosis factor alpha activity and negating the protein therapeutic effects. Now we're going to discuss about the clinical assay for cytokines. Activation for the immune system is a significant component of autoimmune disease such as rheumatoid arthritis, psoriasis, and inflammatory bowel disease. In addition, the role of cytokines in developing an effective immune response to certain infectious disease has been well studied. For example, tuberculosis is controlled by cell-mediated immunity, not humoral immunity, and immunity is therefore driven by Th1 cytokines. Clinical evaluation of the cytokines profile in the patient could be of prognostic and diagnostic value to the physician. Although there are several cytokines assay format available in Europe, not have been approved for clinical use by the FDA at this time. The most basic test is the ELISpot assay, which employs the enzyme-linked immunosorbent assay or ELISA technique on in vitro activated peripheral white blood cells. In this process, either a monoclonal or polyclonal antibody specific for the chosen cytokines is pre-coated onto a microplate. Antigen-stimulated, mitogen-stimulated positive control, or saline-stimulated negative control white blood cells are pipetted into the wells, and the microplate is placed into a humidified CO2 incubator at 37 degrees Celsius for a specified period of time. During the incubation period, the immobilized antibodies in the immediate vicinity of the secreting cells bind the secreted cytokines. After any cells and unbound substance are washed away, a biotinylated polyclonal antibody specific for the chosen cytokines is added to the wells. Following a wash to remove any unbound biotinylated antibody, alkaline phosphatase conjugated to streptavidin is added. Unbound enzyme is subsequently washed away, and a substrate solution is added. A colored precipitated form and appears as spot at the site of cytokine localization, with each spot representing an individual cytokines secreting cells. The spot can be counted with a stereo microscope or with an automated LC spot reader. Multiplex ELISAs utilize several detectors, antibody bound to individual microwells or antibody microarrays, and allow for simultaneously detection of several cytokines from serum. Current formation allow for the detection of 12 to 25 pro and anti-inflammatory cytokines in one reaction. In the microarray format, each well on the slide contains a microarray of spotted antibodies with 4 spots for each of the 12 cytokines, plus additional spots for positive and negative control. The replicate spots allow for acquisition of reliable quantitative data from a single sample. So, new microbead assay allow for the simultaneously detection of multiple cytokines in a single tube. Each bead type has its own fluorescent wavelength, which when combined with the fluorescent secondary antibody bound to a specific cytokines, allows for the detection of up to 100 different analytes in one tube. The use of a multiplex bead array enables simultaneously measurements of representative pro-inflammatory cytokines such as interleukin-1 beta, interleukin-1 receptor antagonist, and interleukin-6, interleukin-8, and tumor necrosis factor alpha. Th1, Th/Th to distinguish cytokines interferons gamma, interleukins 2, interleukins 4, interleukins 5, and interleukins 10. Non-specific acting cytokines are interferons alpha, interleukins 13, 15, and 17, and chemokines IP10, MCP1, MIP1 alpha, and RANTES. One drawback of protein-based technologies is the short half-life of certain cytokines. This can be overcome by looking at RNA expression in cell using reverse transcription polymerase chain reaction. The PCR product is made using a fluorescent label primer and can be hybridized to either solid phase or liquid microassay. Solid phase arrays have up to 40,000 spots containing specific ssDNA oligonucleotides representing individual genes. Clinically useful arrays generally have substantially fewer genes represented. Fluorescence of spots indicates that the gene was expressed in the cell and that the cell was producing the cytokines. The liquid array utilizes the same bead as the antibody microbead arrays discussed earlier. However, instead of antibodies, these beads have oligonucleotides on their surface and allow up to 100 different cDNAs to be identified. The combination of the bead fluorescence and the fluorescence of the label of cDNA produces an emission spectrum that identified the cytokines gene that was expressed in the cell wall, in the cells. Now we're going to summarize overall now. So, cytokines are the functional regulatory proteins for the immune system and are secreted by white blood cells and a variety of other cells. They exert activity modulating effects on cells of the hematopoietic and immune system through the activation of cell-bound receptor proteins. Cytokines are induced in response to specific stimuli such as bacterial lipopolysaccharide, flagellin, or other bacterial products. Through the ligation of cell adhesion molecules or through the recognition of foreign antigens by host lymphocytes. The effects of cytokines in vivo includes regulation of growth, differentiation, and gene expression by many different cell types, including leukocytes, through both autocrine and paracrine activities. Occasionally, cytokines will also exert systemic or endocrine activities. Individual cytokines do not act alone but in conjunction with many other cytokines that are induced during the process of immune activation. The resulting network of cytokines expression regulates the activities of leukocytes by modulating cytokines activities. This led to the elimination of the infection. The combined cytokines produce a spectrum of activities that leads to the rapid generation of innate and adaptive immune responses. In fact, the inability to generate certain cytokines pattern often determines the outcome and clinical course of infection. In certain circumstances, massive overproduction produces a cytokine storm that leads to shock, multi-organ failure, or even death, thus contributing to pathogenesis. The cytokines were originally thought to act solely on cells of the immune system, but we know now that many cytokines also act on cells outside the immune system. Vascular endothelial cells, astrocytes, liver cells, and muscle cells, and many other cell types respond to cytokines. The pleiotropic nature of cytokines activity relates to the widespread distribution of cytokines receptor on many cell types and the ability of cytokines to alter expression of many genes. Several different cytokines may share properties. They activate some of the same pathway in genes. This redundancy may be explained by the sharing of cytokines receptor subunit by many cytokines. However, redundancy of cytokines activities cannot be explained entirely by shared receptor subunit. Tumor necrosis factor alpha, interleukins 1 and interleukin 6 exerts many of the same biological activities, but each utilize a distinct receptor. Here, the cytokines are activating similar immune response pathway through different signaling pathway. The major cytokines involved in the initial stages of the inflammatory response are interleukin 1, interleukin 6, and tumor necrosis factor alpha, and the chemokines. These cytokines are responsible for many of the physical symptoms attributed to inflammation such as fever, swelling pain, and cellular infiltrate into damaged tissues. Engagement of the tumor PCR by the appropriate peptide MHC complex triggers clonal expansion of CD4 T cells, CD4 T helper cells. Naive T cells can differentiate into Th1 and Th2 or Treg cells lineage. With the spectrum of cytokines expressed in the initial response determining the lineage, the Th1 lineage is driven by the expression of interleukins 12 by dendritic cells and is primarily responsible for cell-mediated immunity, while Th2 cells are drive antibody-mediated immunity and developmentally regulated by interleukins 4. Treg cells are derived from interleukins 10, responsive to naive T cells and help to regulate the activities of Th1 and Th2 cells through the activities of interleukins 10 and transformative growth factor beta. The colony-stimulating factor include interleukin 3, erythropoietin, and granulocytes, macrophages, and granulocytes macrophage colony-stimulating factor or GCSF, MCSF, and GMCSF respectively. The colony-stimulating factor are responsible for inducing differentiation and growth of all white cells classes and help to drive the immune response by ensuring that the number and type of cells available are adequate. Anti-cytokines therapy continue to be developed for many pathological conditions such as Crohn's disease and rheumatoid arthritis. These therapies will drive the need for laboratory assays for cytokines and other marker of immune activation.